theory classes, we have learned about the various importance of the red blood cells, what are the functions of the red blood cells and their important role in carrying the oxygen to the tissues, to carry the waste products away from the tissues, as well as various other roles of the red blood cells. And you should keep in mind, and it is always a very well known fact that whenever an individual arrives in the hospital, be it with even simplest of reasons, often the routine blood examination becomes the primary examination that a doctor suggests so as an assistance or as along with other investigations, a routine blood examination is almost always carried out, even if the individual arrives with even simplest of complaints. So it is essential that the routine blood examination be carried out, under which there are various parameters of the blood which are tested, like the total RBC count as we are doing today, total WBC count, differential leukocyte count, hemoglobin estimation, reticulocyte counts. So all of these are under the routine blood examination, because by having the information about these various blood parameters, it will help us in assisting to diagnose a certain condition. So this is the reason why the total RBC count is also one of the very, very important tests that are carried out in the clinical setting. A routine blood examination is always, and mostly in 99% of the cases, the routine blood examination is always advised along with the other tests that might be required, because it plays a very significant role in coming to a significant or to a particular diagnosis about the individual's illness or the ailment. All right, so having said that, I would like to share my slide with you. I have prepared certain slides, and I also have a video at the end so that you can at least know about how the total procedure is done. What does it look like when you are viewing it under the when you are doing it in the practical setting. So just give me a second. All right, so can you see the slides? Students, can you view the slide? All right, so the topic for today's practical is total red blood corpuscles count, or total RBC count. But along with the RBC count, since you have an idea about the Newber's chamber and how the counting on the Newber's chamber is done already, so we will also compare it with the total WBC count, because it is almost the same. It's just that certain things are different, which I will explain to you as we go through the slides. So first and foremost, focusing on the total RBC count for now. So the total RBC count, it is the number of the erythrocytes per liter of the blood. So how many red blood cells are there per liter of the blood is the examination, is what total RBC count is about. So what are the various instruments that we require is the hemocytometer. The hemocytometer, which is usually comprised of a improved Newber's chamber, which you know and you can see here in this picture. And then there is RBC pipette that is required for the total RBC count. And if it is the total WBC count that you are doing, it is the white WBC pipette. There are certain differences between RBC and WBC pipette, as you must know, or you know it already. So just briefly, I'll tell you that there is difference in the marking, first of all, in the RBC and the WBC pipette. So in the RBC pipette, the marking is 0.5 and then 1 and then up to 101, while in case of the WBC pipette, the marking is up to the 11. Similarly, the bead at the center of this enlarged bulb is also different. The bead in RBC pipette is red and the bead in the WBC pipette is white. Similarly, the difference is also with the bore of the diameter, in which the WBC's bore is quite wide than that of the RBC. And if any of this is not visible, then the another important visible feature by which you can distinguish the RBC and the WBC pipette would be the mouthpiece, the color of the mouthpiece, in which the RBC pipette, the color of the mouthpiece is red, whereas in case of the WBC pipette, the color of the mouthpiece is white. So this is how you differentiate the various differentiating features between RBC and WBC pipette. A very commonly asked question in your Viva as well as one of the commonly given instrument for identification in the spotters. So make sure that these various points you look for before you come to a conclusion. All right. So having said that, another important apparatus that we require for this procedure is the RBC diluting fluid. Similarly, for since here the blood sample that obtained for this procedure will be carried out by pricking the finger. So you know that we require a complete set of instruments for the pricking itself, that is the lancet, the cotton wool, as well as the spirit. So, and then we require a compound microscope for the visualization of the Neuber's chamber under the microscope, a cover slip which is usually placed on top of the counting chamber of the Neuber's chamber, and okay, so these are the various instruments, the various apparatus that are required. So number one is for the collection of the sample of the blood, we require a cotton wool, lancet, and spirit. And then of course we require a blood sample, and then we require certain instruments, the Neuber's counting chamber for the counting of the cells, the WBC pipette and RBC pipette for the collection of the for the withdrawal of the blood collectively for RBC count or the WBC count. Similarly, the diluting fluid, and for the respective RBC or the WBC diluting fluid are different. So we will see in details. So what exactly is the RBC dilute? Of all the instruments you've seen RBC pipette, you've seen WBC pipette, you've seen the counting Neuber's, improved Neuber's counting chamber, you've seen microscope, you know what the pricking set for the blood pricking or finger pricking are. But then the new thing here is the RBC diluting fluid. So what exactly is the RBC diluting fluid? Okay, so RBC diluting fluid is actually a fluid that often comes in a bottle, pre-bottled form like this, but it has various composition, and each composition serves a specific function. So if we look at the composition of the HIMS fluid, it is another name for the RBC diluting fluid, and this should also be kept in mind. The name for the RBC diluting fluid is the HIMS fluid. So it consists of certain ingredients like the sodium chloride 0.5 grams, sodium sulfate 2.5 grams, mercury chloride 0.25 grams, and all of these substances are dissolved in 100 milliliter of the distilled water. So it comes in a pre-bottled form. We don't have to collect these individual substances and make a solution out of it. Rather, we get a pre-bottled form called as the HIMS fluid in like in the market. So we buy that and we carry out the experiment. So the sodium chloride here, it is acting and as sodium chloride as well as the sodium sulfate, both of them are acting to provide the isotonicity to the solution. By providing the isotonicity to this solution, it means that the RBC to which it will be diluted with will not be lysed, will not be broken down, will not be damaged. That might hamper the total count of the RBCs. So the main function of the sodium chloride and the sodium sulfate is to maintain the isotonicity of this solution so that the proper structure of the RBCs can be preserved and the counting will be more or less accurate. All right. Similarly, if we are looking at another composition, that is the mercury chloride often acts as an antifungal as well as the antimicrobial agent. So it prevents the contamination of the blood that has been collected by fungus or any kind of bacterium. So it acts as an antifungal as well as an antimicrobial agent. And the sodium sulfate, besides providing the isotonicity to the solution, what it also does is that it also acts as an anticoagulant, meaning that it prevents the blood from clotting. And sometimes it also acts as a fixative, meaning that it preserves the structure of the cells and allows proper fixation of the Neuber's chamber so that the counting can be done easily. So that is about the RBC diluting fluid. Okay. Now we will continue with the procedure. So here what we are doing, the principle of this whole experiment is to facilitate the count of the RBC in a specific volume of the blood, which is diluted with a specific volume of the isotonic fluid, which in this case is called as the RBC diluting fluid or the HIMS solution. All right. So what is the procedure? So first of all is that we have to collect the blood sample. So as shown in the picture, as you can see here, you can also collect, like often the experiments are also done by the blood sample that has been collected previously and has been mixed with an anticoagulant. But in our setting, in our laboratory, we are going to prick the fingers of our hand for the collection of the blood sample. And for this, all of you know the ideal protocols before you carry out the pricking process. You have to just wipe your hand with, so as to prevent contamination, you have to wipe your hand with spirit, and then that is soaked in the, like the cotton ball that is soaked in the spirit, and then you have to make a prick, a quick prick, so as to prevent the deep puncture of the finger. And the 3 fingers as shown here should be used for the pricking. The thumb and the little finger should not be used because of their connection with the palmar arch of the hand, because any kind of deep pricking that might result in a deep wound might cause the unwanted or severe infections throughout the hand. So in order to prevent, so these are very sensitive areas, so you should avoid pricking thumb as well as the little finger. So these 3 fingers, the finger apart from the thumb and the little finger should be utilized for the collection of the blood sample. So immediately after you collect or you form a drop of blood on the surface of the finger following pricking, what you should do is you should use the RBC pipette to suck this blood up to the mark of 0.5. So you know that in case of the RBC pipette, there is a marking, there is 0.5, there is 1, and then there is 101. So you have to draw the blood up to 0.5 mark. And for to be more, not to prevent the error from happening, you can draw it slightly more than 0.5, and then later you can just tap it off into a filter paper and bring it up to the 0.5 mark. So once you have drawn the blood up to the 0.5 mark, so it will be clear once you see the video also. So for now, just try to imagine these things happening in the lab inside your mind only. So once you draw the blood up to the 0.5 mark, because we are using the fresh blood here, you should always keep in mind that it can coagulate quite quickly. So what you have to do is you have to be a little bit quick in your steps so as to prevent the clotting of the blood inside the pipette. So once the blood has been adjusted up to 0.5 mark, so you have to suck up the RBC diluting fluid up to the 101 mark. So that's to prevent the blood from clotting. So once you've drawn this RBC diluting fluid to 101 mark, now you have to remove it from you. All of this you are doing by sucking it with your help of the mouth. By putting the mouthpiece in your mouth, you're sucking the blood up, and then you're sucking the RBC fluid up. So once the RBC fluid has been sucked up to 101, you have to immediately remove the mouthpiece from your mouth, and you have to place it horizontal on your hands. Okay, so place it horizontal on your hands. Make sure that you're not keeping it at an angle, because that way the fluid will flow out from the tip of the pipette only. So you have to place it horizontally, and then you have to just shake it in a rolling motion. Actually, you have to just shake it by keeping it at a slightly right angle to the long axis, and then you have to just roll it along your palm of the hand so that there is proper mixing of the RBC diluting fluid with the blood. Okay, so this procedure has to be carried out for about 2 to 4 minutes. Okay, in your book it is given certain seconds. Did you receive manuals or not, by the way, students? Do you have manuals with you right now?
No, ma'am.
No, you were not given, or you didn't take it along with you?
Ma'am, we didn't take it along with us.
Okay, all right. So, but you've been given. Okay, all right. So once you, it's very difficult, I understand, but yeah, please try to follow me. But then we'll have a demonstration video at the end also. So what you have to do is, once it has been sucked up to 101 mark should be removed from your mouth, and it should be placed flat on the palm of your hand, and then you should roll it gently so as to facilitate the proper mixing of the diluting fluid with the blood. So once you've done this for about 2 to 4 minutes, then you can be sure that the RBC diluting fluid has been properly mixed with the blood, and then after that comes the process which is called as the charging of the Neuber's chamber. So we know that once we've sucked up the blood up 0.5 millimeter and then we suck the fluid, so we know that the fluid that has been sucked at the last moment, that does not contain any blood mixed with it, so we discharge that few drops from the stem. So once we've discarded those few drops from the stem, the diluting fluid from the stem that has not been mixed with the blood, now we are ready to charge the Neuber's chamber. So charging is actually nothing, but it is just the loading of the Neuber's chamber with the RBC diluting fluid mixed RBCs. So what you have to do is we have to put a cover slip in the place of the on top of the hematocytometer, and then by holding the pipette at a slight angle, you can see here. So you have to put it at a certain angle. You can see that there is a Neuber's chamber here. The Neuber's chamber has been put on top of it. It has been covered by a cover slip, and then this is the RBC pipette, and then you can see that by keeping at a certain angulation, you have to just touch the point where the cover slip touches the Neuber's chamber. So by just touching with it, what happens is that the fluid that is present within the RBC pipette will uniformly flow on the surface of the chamber by the capillary action, and this process is called as the charging of the Neuber's chamber. Understood? Sometimes what happens is that if a big droplet of this fluid forms on the tip of the pipette, then it might result in the overcharging of the chamber, meaning that if there is a big fluid that is formed at the tip of the pipette, then what happens is that this fluid will immediately spread along the surface of the chamber, but sometimes it might even overflow inside the trough. So if this happens, then what you have to, this is called as the overcharging. And what you have to do is you have to actually remove this chamber, you have to remove this cover slip, you have to wipe the entire, or you have to completely clean the Neuber's chamber, and a fresh drop has to be taken. Similarly, sometimes if you fail to make an adequate drop, then the fluid might not spread throughout the chamber, like the counting chamber, and that is called as the undercharging. So in that case also you have to carry out the entire procedure again. So you have to make sure that the charging is done appropriately. So you have to make sure that there is not particularly this much drop, but you'll kind of get an idea once you start doing. So you have to make an adequate, there has to be an adequate drop of this fluid that has to be used to charge the chamber properly. So once you have charged this chamber properly, now what we have to do is we have to load this charged chamber into the microscope. So once you've loaded it onto the microscope, as always, it is always required that you always start viewing from the low power magnification. So at first, what you have to do is you have to set the microscope to a low magnification, and then you have to view the Neuber's chamber, and this low magnification is to make sure that the spread of the cells are even. And after making sure that you've focused on the chamber for counting the RBCs, and once you've made sure that the cells distribution is even, then you can adjust your microscope lenses to a higher magnification power, to a greater magnification power, after which you can, that is switching to a 40 objective lenses, and you can start counting the squares. So all of you know that within the Neuber's chamber, this is what you've seen this before, yes? Yes or no? This has been explained before. Yes?
Yes, ma'am.
Students, yes or no? Please respond,
yes,
okay? Anyways, it's difficult for me to teach like this, and if you don't respond, and who is this placing yikes? Yikes on as their profile picture? Very insulting. What is yikes? And my eyes are just focused on that, yikes. Okay, whatever. So you know that you've seen this is what the counting chamber looks like when you focus it under the microscope. So this light blue chamber, they are for counting the white blood cells. So this light blue, this upper left, lower left, upper right and lower right are utilized. This light blue colored chambers are for counting the WBCs. And now since we are focusing on counting the RBCs, so this central big square is the focus point for counting the red blood cells. And you know that within this central square also, it is further divided into total of the 25 squares. And this 20 out of this 25 medium-sized squares that is present here, then only these five medium-sized squares are used for counting the RBCs. These red colored areas are the areas that are used for counting of the red blood cells. Okay, so this is what a Newber's chambers looks like. So it has been divided into several squares. The upper four squares, the upper left, the upper lower left, the upper right, the lower right are used for counting the WBCs, and the central big square that is present there, that is utilized for counting the RBCs. Even in the central square, it is again divided into 25 medium-sized squares. Out of those 25 medium-sized square, only the 5 squares are utilized for counting of the red blood cells. And of these 5 squares that are utilized for counting the red blood cells, those are the upper left, lower left, upper right, lower right, and the central square. So this is how the RBC, this is where the RBC is counted within the Newber's chamber. So if you focus it under the microscope, it looks like this. So of this is the central square. This is the central square here, which is used for counting the red blood cells. And among this central square, only this 5 squares, total of 5 squares are used for counting the red blood cells. So you can see this is what it will exactly look like. These are the cells that are within, and these cells has to be counted. And you know that the square that has to be counted, the square that has to be counted is that the cells that touch the top and the left hand borders, isn't it? The top and the left hand borders they are also taken, whereas this those that are excluded, the rest two borders are excluded. These are not counted. The cells that are touching here, they are not counted. Only the cells that are touching the top border and the left border, they will be counted. And you will count in all these 5 squares. After counting all these 5 squares, then you will sum up the total number of cells. And since the erythrocyte count is to be the unit is, you are counting it in volume, cubic millimeter of the blood, then you will find out we'll see how it is counted. So for now, I hope you understand how it looks like. So this is exactly what it looks like when you are viewing it under the microscope. So once you have loaded your Newber's chamber and you have viewed it under the microscope, now you are to count the cells present in all these 5 chambers. Okay. So once you have done that, now what you are going to do? So how are you going to calculate the volume? How are you going to calculate the total number of the RBCs? Is now what we are going to study. Okay, it is simple maths. You just have to understand. So you know that what is the total? What is the length of the 1 square of this Newber's chamber? What is the total length of this side? Say this side. What is the length? You know this. What is the length of 1 square, of 1 big square? What is the length of 1 big square? 1 millimeter, isn't it? Yes. So the length of one side of the 1 square millimeter, it is 1 millimeter. So this whole, if this is the RBC counting chamber in focus, then we know that the length of this square is 1 millimeter. So how we are going to calculate is that So there are certain things that you have to know to calculate. Number one is that you have to at first count all the number of the cells in all the 5 blocks of the 16 smallest squares. So you have to count the cells in each of the 5 blocks that is required for counting the RBCs. And then you have to calculate the dilution factor. You have to know the dilution factor of the fluid that you have taken. So you know that of the total amount in the RBC pipette, initially the volume of the blood that you took was 0.5, isn't it? You took 0.5 of blood, so the volume of the blood that you took was 0.5, and the total amount of the fluid that you took was 100, because you filled it, sucked it up to 101. But while you sucked it up to 101 and 1, the 0.5 has been already attained has been taken up by the blood, and the 0.5, the remaining, you tapped it away because that was not diluted with the blood. So about 0.5 you discarded. So the total volume of fluid, the fluid only that you have is 100. So the dilution factor here is 200. Are you following me or not? Yes or no?
Yes, ma'am.
Yes, so the dilution factor is 200. So now that how we are going to calculate is that now we have to calculate the volume of the fluid that we have examined. So we know that the side of the one large square is 1 millimeter. Yes. So if we are looking at the side of one medium square in which you have calculated the RBC. So what will be the side of this small? What will be its size of this square that you have calculated? It will be 1/5 millimeter. Yes or no? Because
yes,
total, yes, the total squares, there are total of 5 squares. So of that, one medium square will have a length of 1/5 millimeter. So if the side of one or the length of one medium square is 1/5, then you know the area. What is the area? Is 1/25, isn't it? Area is L square is area. So the total area of one medium square would be 1/25 square millimeter. You know how to calculate area. So how many squares have you counted? You have counted 5 squares. So the area of 5 squares would be 5/25. That is 1/5 square millimeter. Yes or no? Yes?
Yes, ma'am.
Yes. Now that you have calculated that, now we have to calculate the volume. So for the calculation of the volume, we know that the calculation of the volume is length into breadth into height, or in this case, depth of the new burst chamber. So you know the area, that is 1/5, and then you know the depth of the new burst chamber, that is 1/10. So the volume is going to be 1/50 cubic millimeter. Yes or no? So it means that the total number of the RBCs in this 5 squares, the volume of these 5 squares, that is 1/50 cubic millimeter, if we assume it to be N, okay, then the number of cell in 1 cubic millimeter of the blood would be 50N, isn't it? By simple mathematical calculation. This you can carry in by arithmetic method. So if total number of RBC in 1/50 cubic millimeter is N, we assume, then we know that the number of cells in 1 cubic millimeter would be 50N. And we have the dilution factor. The dilution factor, we have found out that it is 1:200. So it means that 1/200, we have diluted the blood. So 1 out of 200 diluted blood contains 50N number of cells, isn't it? So the undiluted blood would obviously contain about this amount of the cells. Yes or no? So it is 200 into 50N cells per cubic millimeter of the blood. So it is, whenever you are asked to carry out the RBC count, the final formula would be, it is N, the total number of cells that you have counted. The N includes the total number of cells in the 5 squares that you have counted into 1,000. Sorry, into 10,000. Yes or no? And that is how you are going to get your volume of the RBCs per cubic millimeter of the blood. Understood or not?
Yes, ma'am.
Yes, so yes, ma'am.
Yes.
I hope you've understood. So this is how you're going to do the RBC count. So I want you to watch a video right now, okay? And then you will hopefully understand what it is about. And then again we will discuss how it is different from the counting of the white blood cells. Okay, so this video is actually clearly depicts the second. We have to do something here. Okay. Let me know if you hear the volume or not, okay? Because most of the times the volume is not heard. Do you see the video opening or not?
No, ma'am.
Okay, just give me a second. You can't see it? Can you see it or no?
No, ma'am.
No? Okay, just give me a second. Just give me a second. How now? Now can you see it? Or no?
Not yet, ma'am.
Not yet. Okay.
There was audio but no video.
There was only audio. Okay, just a second.
Excuse me, ma'am. You can upload it on Microsoft Teams and we can visualize it offline.
Oh,
Right now.
I could do that, but for now I just wanted it to be. I don't. But yeah, I could do that too. Just give me a second. Okay, so what I will do is I will upload it, okay, so that you can see it. So sorry, okay, I just had to. I don't know why. Some time. Okay, so I've uploaded it. So could you please all open it, okay? And then you view the video and then see how it is done. And then we will come back to how WBC counting is done. Okay, just give it a few seconds. It will upload. It's uploading. Okay, so can you see it in your in the physiology in Teams?
Yes, ma'am.
Yes. So open it and then view it. So have you all found it? Yes?
Hello everyone. Today we'll discuss about total RBC count.
Can you also,
In total RBC count,
can you?
a sample of blood is diluted 200 times. And the
No,
cells
ma'am.
are counted.
No, it has not been uploaded. Huh?
For RBC count, we require RBC pipette.
You can't view it.
No, ma'am. We are just listening the audio.
No,
The video has not been uploaded.
yes, ma'am, it is uploaded.
It is uploaded, isn't it? Is it visible or not?
Yes, ma'am, it's visual.
Okay, so all of you, please go to the physiology teams, in the teams in physiology, and see the link of the video that I've posted. I think the video itself has been uploaded there. So please, right now, go through it because I'll be asking questions based on that. So please make sure that you go through it. Any of you having any problem or not?
Hello everyone. Today we'll discuss about total RBC count. Here, in total RBC count, a sample of blood is diluted 200 times and the cells are counted in the counting chamber. Starting with the material, for RBC count, we require RBC pipette. Here, this is the RBC pipette. We require improved mumbaI chamber and cover slip. Microscope, compound light microscope we are using.